Hello, hi. I'm Debarpita, and this work was done in collaboration with Rohini Balakrishnan and CES IISc. And let's take a quick look inside inside a tree cricket's mating game. So, sorry. Yeah. So acoustically signaling species, most likely males of signaling species, signal to attract conspecific females. And when a female perceives a conspecific male call, she performs a directional movement known as phonotaxis. Now, the same signal produced by the male can be perceived by a conspecific male, and that sets the stage up for male-male competition. Now, with that, a male can have different responses, and these responses can be satellite strategy, where a male decreases its energetic investment, stops calling, and move towards a conspecific caller to intercept a female performing phonotaxis towards the acoustic host. A male can also show endurance compression or acoustic compression if he increases his loudness or chirps faster, and this would indicate fitness to females and kind of intimidate males. However, males could also avoid all sorts of competition, stop calling, and move away. Even though these expectations seem very naive, some of them were found in literature, such as the satellite strategy by Alter and Zook, in the literature by Alter and Zook, and where male field cricket showed strong phonotaxis towards other calling male, which kind of indicated the satellite strategy. In another study by Wild et al., they found that inhibition was something in males where male field crickets in the presence of more males called only when they were further away from experimental males. In aggregations of katydids, they also found acoustic compression where the central male called faster upon perception of peripheral males, and when these peripheral males were removed, they called slower. Now with that, I studied the Acanthus-Henry system, which has three mating systems, such as the mating strategies, such as the calling, the baffling, where a male cuts a hole into a leaf and calls through it to amplify its sound, and it's an alternative mating strategy, and the satellite strategy. And as these strategies are very flexible in these males, that's why this system was very good to study the sexual selection and competition. Now with that, the main questions we asked was, what do males do when they perceive a conspecific call, and does it matter when they perceive this conspecific call, and when in the study was very important, and you'll see why. Now, the conspecific call in this study was baffling males playback, and that can get 8 to 12 decibels louder than a normal calling male. However, in our study, we set it to 70 decibels as a fixed. Now, with the experimental setup, we had a tea maze which was built out of stripped down Hyptis cervulens shrub, and because this is a host plant to the this cricket, and on either side of the tea, we had a speaker which was randomized to play a conspecific call at 70 decibel and at the center of the tea, which was the choice point, and that's where we placed a leaf of Hyptis cervulensis, and where a male could go up and start calling. Now the entire setup was inside an anechoic chamber, and the anechoic chamber was thermoregulated to 25 degrees Celsius. Okay. Now we had two experiments. The first experiment was where we released a male and let it first start calling, and then they heard a conspecific call. And the second experiment is where we released a male and immediately the male heard a conspecific call before they could start calling themselves. Now, all results in this study was done by comparing and contrasting different portions of these experiments, and with that, the first question we try to answer was does the call propensity and latency change with playback? And call propensity in this case was the number of males who called within the playback period, which is like for the first it is 10 to 20. The dashed line basically, and in the second experiment the first 10 minutes was the playback period, and the latency was when the male first started calling. Now with that, we found that the call propensity was very different for males who heard the playback at the start, and it was very low, and simultaneously the latency of calling had shifted in time for the same group of males. Now, the second question we wanted to answer was, do males move differently when they hear a louder playback after they start calling? Because we are seeing that these males are remaining silent. So what are they doing when they are remaining silent? And for that, we just looked at the movement during the playback period for both the experiments. And interestingly, we found that males who heard playback at start moved more. Furthermore, they moved towards a conspecific call, which was a speaker emitting the conspecific call. And then we just wanted to test, do these males compete acoustically? And we use call characteristics such as sound pressure level, which stands for loudness, chirp rate, which stands for how fast the male is calling, and the call effort. Now for this we had experiment 1 and a control. So in experiment 1 we released a male and gave it 20 minutes to start calling. And let's say the male in our study starts calling at 5 minutes. So we gave it 10 more minutes to keep calling so that the call stabilizes. And within this 10 minutes of calling, at 5-minute point, we measured the sound pressure level and the chirp rate. And after that, a 5-minute window, we measured the call effort. And then it heard a conspecific playback of three different exposure types, which was the 2 minutes, the 5 minutes, and the 10 minutes. Now, immediately after the playback stopped, we measured the sound pressure level, chirp rate, and 5 minutes of calling effort. For the control, we did the same thing except there was no playback at all. Now, for sound pressure levels, we found there was no change in sound pressure levels as a response to playback or with time. However, for chirp rate, we saw a change in 2 minutes exposure and 10 minutes exposure. And when we also saw the sound pressure, the chirp rate changing for control, we could only say that it changes with time, and the longer the male keeps calling, the faster it gets. Now, call effort also did not have a response to playback, or it was not a function of time. Now, on the whole, coming to the summary is, many studies have actually explored response of conspecific males to conspecific calls, but however, they have missed the condition or behavioral state dependent response of these males, which we cover in the study. And so what we find essentially is time of playback affected calling propensity and the latency also. And with changing latency, we can say that we saw some inhibition of calling, and the same males who heard the playback before they could start calling moved actually towards the conspecific playback, and this could show the existence of satellite strategy. However, we cannot rule out that it could also be aggression, even though there are telltale signs of aggression such as body rocking or aggressive calling. And thus it mattered when the male heard a playback. However, we did not see any evidence of acoustic compression with changes in sound pressure level, chirp rate, and calling effort. However, chirp rate changed with time, which was new to us, and we did not find in any other literature. So essentially what this study showed was males have unique strategies, condition-dependent unique strategies, which they use to outcompete rival males to increase their own fitness. With that, these are the people I would really like to thank. Thank you.
So, a few questions.
Thank you very much. Very interesting. Did you find any among individual male differences in their chirp rate, irrespective of, the treatments and so on?
So, like, you mean like a variation?
Like, were there some males that chirp faster than others consistently?
Yeah, there were. So I could go back. Yeah, so, I mean, you can see some males actually chirp faster than the others, but I mean, they also change with time. So even if they start chirping at a particular point, with time they're increasing their chirp rate.
Okay, but as a statistical test of whether there's actually among individual variation in that, other than just that there is you
We didn't do any,
Yeah.
to see the variation.
Yeah, okay, I think it'd be interesting because you have the data to do that. Thank you very much.